wanted in. Oh darling, get me drunk and make me feel it's not my fault, I'm not to blame. These ain't my sins, I broke my chains. There's more to do and I still want to live.
I feel your breath upon my neck, a soft caress as cold as death.
Cold as death.
I feel your heart beat in my soul, our futures bound, our bodies know. Your blood like wine, I want it in. Oh darling, get me drunk, invite me. It's not my fault, I'm not to blame. These ain't my sins, I broke my chains. There's more to do if I can only leave. I can't go yet, don't let me die. I'll never stop until I'm done. But just tonight, maybe I'll rest in peace. I feel your breath upon my neck, a soft caress as cold as death. I hear your heart beat in my soul, our endings bound, our bodies know. I can't go yet,
Don't let me die,
I want to live.
my only one. There's more to do if we can only live. The clock won't stop and this is what we get.
Hello, hello, and welcome to another day of studying. Today, in Advanced Pathophysiology, we're going to talk about cancer's biology. We're not going to read all of it though. I only have a certain section here to read. I know our pages here may not line up perfectly, but it's going to be from 1122 on this PDF to 1154 before we jump into cancer's epidemiology, where it's only going to be a brief portion of that chapter as well. Pages 1212 to 1231. This little section is going to disappear. I'm going to keep it here on the side so I can see it for reference, but other than that, we'll jump right into it. Cancer is a leading cause of suffering and death in the developed world. Over the past 35 years, intense research has led to a significant enhanced understanding of this complex and frightening disease. It is now understood that cancer is a collection of more than 100 different diseases, each caused by a specific and often unique age-related accumulation of genetic and epigenetic alteration. Environment, heredity, and behavior interact to modify the risk of developing cancer and the response to treatment. Improvement in treatment strategies and supportive care, coupled with new, often individualized therapies based on advances of the basic pathophysiology of malignancy, have contributed to an increased number of effective options for these diverse, often lethal, disorders collectively called cancer. So, jumping right into it, cancer characteristics and terminology. Again, in our study booklet here, we'll have a whole list of cancer biology, and a portion here is called Name the Neoplasm. So when we come across one, we'll make sure to tick it off our list. Any discussion of cancer must start with a definition of what it is and what it is not. Although most readers may have an intuitive understanding of the disorder, composing an exact definition that encompasses this broad category is more challenging. The term cancer derives from the Greek word crab, or carcinoma, which the physician Hippocrates used to describe the appendage-like projections extending from tumors. The word tumor originally referred to any swelling that was caused by inflammation, but is now generally reserved for describing a new growth, or neoplasm. So we know that neoplasms are new growths. Resulting from an abnormal growth following uncontrolled proliferation and serving no physiologic purpose. Not all tumors or neoplasms, however, are cancer. The term cancer refers to the malignant tumor and is not used to refer to benign growths, such as lipomas or hypertrophy of an organ. So a lipoma is a benign a lipoma is benign growth, but it doesn't actually specify what kind of benign growth, whether it's fat cells, smooth muscle cells, or skeletal muscle. So we'll question mark that because it could, I guess, technically be in any of those sections. The National Cancer Institute, NCI, of the National Institute of Health, the NIH, defines cancer as a disease in which abnormal cells divide without control and are able to invade other tissues. The definition of benign versus malignant are present in the following text and shown in figure 12.1. Okay, so here we're taking a look at a uterus, comparison between a benign and a malignant tumor of the same origin, from Kumar, Robinson, the Cochrane Pathologic Basis in Philadelphia in the Saunders book. We have a benign tumor, a leiomyoma, and a malignant tumor, a leiomyosarcoma. All right, so tumor classification and the nomenclature. Proper identification of a cancer is important for many reasons. Different cancers will have different causes, different rates and patterns of progression, and different responses to treatment. The classification starts with knowing the tissue and organ of origin, the extent of distribution to the other sites, and the microscopic appearance of the lesion. Increasingly, it also includes a detailed description of the critical genetic changes in the cancer. Benign tumors, which are not referred to as cancers, are usually encapsulated with connective tissue and contain fairly well differentiated cells and well organized stroma. Again, we can see that here in the picture. Right here. They retain recognizable normal tissue structure and do not invade beyond their capsule, nor do they spread to regional lymph nodes or distant locations. Mitotic cells are very rarely present during microscopic analysis. Benign tumors are generally named according to the tissues from which they arise, with the suffix of "oma." which indicates the tumor or mass. For example, a benign tumor of the smooth muscle of the uterus is a leiomyoma, and a benign tumor of fat cells is a lipoma. So a lipoma is a benign tumor of the fat cells. Benign tumors of the colon or stomach, usually present as colonic or gastropolyps, and those are melanocytes, present as dark-colored nevi or birthmark moles. It is important to understand that benign tumors can become extremely large and, depending on their location in the body, can cause morbidity or be life-threatening by compressing normal tissue, preventing blood flow to the region, ischemia, or causing necrotic death of normal tissue. For example, a benign meningioma at the base of the skull may cause symptoms by compressing adjacent normal brain tissue. Benign tumors of endocrine organs may lead to overproduction of hormones. Some tumors, initially described as benign, can progress to cancer and then are referred to as malignant tumors, which are distinguished from benign tumors by more rapid growth rates and specific microscopic alterations, including loss of differentiation and absence of normal tissues. We're going to take a look at figure 12.2 in just a second. One of the microscopic hallmarks of cancer cells is anaplasia, the loss of cellular differentiation. Malignant cells are also pleomorphic, with marked variable size and shape. They often have large, darkly stained nuclei. Mitotic cells are common, and malignant tumors may have a substantial amount of stroma, but it is disorganized with loss of normal tissue structure. Malignant tumors lack a capsule and grow to invading nearby blood vessels, lymphatics, and surrounding structures. The most important and deadliest characteristic of a malignant tumor is their ability to spread far beyond the tissue of origin, a process known as metastasis. So here we're taking a look at a normal tissue cell. Here it is benign because it's encapsulated and doesn't go further than that. A well-differentiated malignant, so I guess it's well differentiated in the fact that it stays within spot, but it's growing malignant outside or further. Poorly differentiated, let's make this easy on the eyes, boom, boom. Poorly differentiated malignant, anaplastic malignant, and then again, benign. So, unlike benign tumors, which are named related to the tissue of origin, cancers are generally named according to the cell type from which they originate. Cancers arising from the epithelial tissue are called carcinomas. Okay, that's definitely on our little question mark here. A malignant tumor arising from the epithelial tissue, carcinoma. Got it. And if they arise from or form ductal or glandular structures, are named adenocarcinomas. So if it's from a gland, it is called an adenocarcinoma. Got it. Hence, a malignant tumor arising from the breast or glandular tissue is a mammary adenocarcinoma. Whereas an example of a benign breast tumor is a fibroadenoma. Oh my. All right. Cancers arising from the mesenchymal tissue, including connective tissue, muscle, and bone, usually have a suffix of sarcoma. So, for example, malignant cancers of skeletal muscle are known as rhabdomyosarcomas. Rhabdomyosarcomas. But these are malignant tumors of skeletal muscle. So we'll check that one right off. Going through our little word list quite quickly here. Cancers of the lymphatic tissue are called lymphomas, whereas cancers of blood-forming cells are called leukemias. However, many cancers such as Hodgkin's disease and Ewing sarcoma are named for historical reasons that do not follow this nomenclature convention. Carcinoma in situ, often abbreviated CIS, refers to a pre-invasive epithelial tumors of glandular or squamous cell origin. Cancers develop incrementally, and they accumulate specific genetic mutations. Careful surveillance for cancer often detects abnormal growths in epithelial tissue that have atypical cells and an increased proliferation rate compared with normal surrounding tissue. These early-stage cancers are localized to the epithelium and have not penetrated the local basement membrane or invaded the surrounding stroma. Based on these characteristics, they are not malignant, but are often called CIS, or sarcoma in situ. CIS occurs in a number of sites, including the cervix, skin, oral cavity, esophagus, and bronchus. In the glandular epithelium, in situ lesions occur in the stomach, endometrium, breast, and large bowel. In the breast, ductal carcinoma in situ fills the mammary ducts, but has not progressed to local tissue invasion. CIS lesions can have one of the following three fates. They can remain stable for a long time, they can progress to invasive and metastatic cancers, or they can regress and disappear. CIS can vary from low-grade to high-grade dysplasia, and with a high-grade lesion having the highest likelihood of becoming invasive cancers. The time that such pre-invasive lesions remain in situ before becoming invasive is unknown. Some carcinomas of the cervix appear as pre-invasive lesions in situ for several years before they progress to invasive carcinoma and metastatic tumors. Knowing how to best treat low-grade CIS lesions is challenging because the proportion that progress to cancer versus the proportion that will never cause clinical problems is usually not known. Although most persons prefer removal of CIS as opposed to watchful waiting, this topic continues to be a source of great debate. So here we have normal epithelium. We have this low-grade intraepithelial neoplasm. So we see that growth here. And then here we go straight to the high-grade intraepithelial neoplasia. We still have some of the norm, but even there it's becoming clustered. And then here it's a straight up invasive carcinoma. A little far. The Biology of Cancer Cells. In two seminal publications, Dr. Douglas Hanahan and Robert Weinberg describe what they considered the hallmarks of cancer. Both articles stimulated considerable discussion and especially debate. The original publication contained 6 hallmarks, but with time and new research findings increased to 8 hallmarks and 2 traits that enable cancer progression. Their analysis remains the leading overview of why a cell is malignant. The following discussion is organized in the context of those 10 hallmarks and enablers. Figure 12.4. 2 fundamental concepts are the foundation for understanding the biology of cancer. Cancer is a complex genetic disease, and the microenvironment of a tumor is a heterogeneous mixture of cells, both cancerous and benign. So, the hallmarks of cancer: evading growth suppressors, enabling replicative immortality, tumor promoting inflammation, activating invasion of metastasis, genetic instability, or a mutator phenotype, inducing angiogenesis, resisting cell death, deregulating cellular energies, sustaining proliferation signaling, and avoiding immune destruction. Interesting. Cancer is a disease of cumulative genetic changes during aging. The fraction of individuals who develop cancer increases dramatically with age. Genetic changes may occur by both mutational and epigenetic mechanisms. Mutation generally means an alteration in the DNA sequence affecting expression or function of a gene. We'll take a look at figure 12.5. Mutation includes small-scale changes in DNA, such as the point mutation, the alteration of one or few nucleotide base pairs. This type of mutation can have profound effects on the activity of resultant proteins. Chromosome translocation are large changes in chromosome structure in which a piece of one chromosome is translocated to another chromosome. Gene amplification is the result of a repeated duplication of a region of a chromosome, so that instead of the normal 2 copies of a gene, tens or even hundreds of copies are present. Gene expression also may be altered indirectly by the epigenetic effects including DNA methylation, histone acetylation, or altered expression of non-coding RNA. Some mutations, referred to as driver mutations, drive the progression of cancer. There may be as many as 140 different driver mutations, although some are more critical than others, and a relatively small number of these are present in an individual cancer. Not all mutations in cancer contribute to the malignant phenotype. Some are just random events and are referred to as passenger mutations. They are just along for the ride. After a critical number of driver mutations have occurred, the cell becomes cancerous. This cancer cell has a selective advantage over its neighbors. Its progeny can accumulate faster than its non-mutant neighbors. This is referred to as a clonal proliferation or clonal expansion. We'll take a look at 12.6 here in just a second as well. As a clone with a mutation proliferates, it may become an early-stage tumor. For example, a carcinoma in situ or benign colonic polyp. The increasing rapid cell division and impaired DNA repairs mechanisms of cancer cells result in a continuing accumulation of mutation through the progression to the most aggressive metastatic lesion. Thus, malignant transformation, the process by which a normal cell becomes a cancer cell, is directed by the progressive accumulation of genetic change that alter the basic nature of the cell and drive it to malignancy. The process of tumor development is a form of Darwinian evolution. Cells with a heritable change that confers a survival advantage outcompete their neighbors. Each cancer cell may develop its own set of mutations, resulting in a genomically heterogeneous mixture of cancer cells, with subsets that have accumulated more and more mutations that increase the cell's malignant potential. The degree of intratumor mutational heterogeneity may vary considerably among tumors. For instance, melanomas have a rather high degree of genetic homogeneity, whereas pancreatic cancers are more heterogeneous and appear to arise from independent mutational events in the pancreas. Thus, cancer cells do not accumulate a critical set of mutations, lose the competition, and die during this process. So this is here figure 12.5. We got a chromosome here. We're looking at the promoter or proto-oncogene. And I guess there are points of mutation, then gene amplification, overexpression, a chimeric protein, and chromosomal translocation. So section 8, the point mutation, that alter one or a few nucleotide base pairs cause the production of a protein that is activated as a result of an altered sequence. This amplification of the cellular gene resulting in higher levels of protein expression. Hence we have our NMYC and neuroblastoma, and then section C here, the chromosomal translocation that are either led to a juxtaposition of a strong promoter, causing increased protein expression, or produce a novel fusion protein that is derived from gene fragments, normally present on different chromosomes. So that could be, for instance, the BCR-ABL in chronic myeloid leukemia. And then our other little picture, 12.6. It looks like we have a normal colonocyte. Then we have our first mutation, a second mutation, and finally a third mutation, which ends up leading us to a mutation or malignant cell. So if our first mutation or genetic event is the inactivation of APCs, cells seem normal, but is predisposed to proliferate excessively. Then if our second mutation is the mutational activation of the KRAS, the cell begins to proliferate too much, but is otherwise normal. With that third mutation, the loss of the DCC overexpression of the COX-2, we end up getting cell proliferates more rapidly. It also undergoes structural changes. So then finally, with that structural change in place, the loss of TP53 activation of telomerase in genetic event 4, the cell grows uncontrollably and looks obviously abnormal. So we see that there in our little structure. We get an event or a mutation, and yes, we still have some normality left, but future generations will continue to change and mutate and change and mutate until it's malignant. So the process occurring during the development of cancers are, in many ways, analogous to wound healing. The initial proliferation of cancer cells and enlargement of a tumor elicit the synthesis of pro-inflammatory mediators by the cancer cells and adjacent non-malignant cells. As with wound healing, mediators recruit inflammatory immune cells, or primary T lymphocytes and macrophages, but also B cells and neutrophils. The cells normally associate with tissue repair, fibroblasts, adipocytes, mesenchymal stem cells, endothelial cells, and parasites. These cells form the stroma, or tumor microenvironment, that surrounds and infiltrates the tumor. We'll take a look at figure 12.7. In some conditions, stromal cells may make up to 90% of a tumor's mass. Extensive paracrine signaling among the stromal and cancer cells affects both populations. Cancer cells increase proliferation and become more heterogeneous during tumor growth, and several populations of stromal cells undergo evolution to phenotypes that promote cancer progression and metastatic potential. Cancer heterogeneity arises from ongoing proliferation and mutation. Tumor-associated endothelial cells, fibroblasts, and inflammatory cells develop different and distinct gene expression profiles, with unique cell surface molecules and patterns of secreted molecules. During this process, there is generally a great deal of cancer cell death, but the surviving cells are more aggressive and many take on a metastatic phenotype. Because continuing somatic mutations may be random, cancer cells in different regions of the tumor may be genetically diverse. Additionally, a population of cancer stem cells may arise, the origin of which is still unclear. Many of the hallmarks of cancer are consequences of cancer-stromal interaction. So here we're looking at preventing tumor growth. Normally, the cell cycle includes that the Ag recognition causes a type 1 cytokine, which I guess attaches to the Ag portion of the cell and triggers apoptosis. However, when there is immune evasion, such as hypoxia, inflammation, angiogenic switch, macrophages, polarization switch, are they reversible? You have a whole mess of things going on. I wouldn't even begin to see how all this works. Your EGF versus the CSF1, immune suppression, the inhibition of cytolytic granule release, your M1 phenotype blocked, tumor-associated Ag presentations are inhibited, the inhibition of B and T cell responses, the overall mobilization, angiogenesis, influences of TAMs, influences of CAFs. I mean, cancers live in a complex microenvironment. Yeah, we can definitely see that. If we look at all the little sections here, your tumor, or, oh my goodness, the anti-tumor tumorigenic macrophage, normal fibroblasts, are T cells and B cells, are ECM, are lymphatic vessels, our CAFs are pro-tumorigenic macrophage, our T regulator cells, our tumor cells, MDSCs, natural killer cells, our dendritic cells, and blood vessels. So cancer cells express tumor-specific antigens that ideally can be recognized by cells of the immune system and inflammatory system. So that's why our natural killer cells, anti-tumor M1 macrophages, T1, T cytotoxic cells, and destroyed by apoptosis or undergo growth suppression by a type 1 or type I cytokines. However, successful cancers produce a variety of cytokines and chemokines that are chemoattractants for stromal cells that infiltrate the tumor and undergo a change to pro-tumor phenotypes. These include tumor-associated M2 macrophages, or TAMs, cancer-associated fibroblasts, or CAFs, mesenchymal stem cells, MSCs, and immune suppressor cells of T cell origin, so T regulatory cells, and myeloid origin, or myeloid-derived suppressor cells. Through multiple receptor-mediated interactions between other stromal cells and the cancer cells, the stromal cells as well as the cancer cells collectively produce a battery of additional cytokines. TGF-beta, type 2 cytokines, and chemokines, or CXCL5 growth factor, so VEGF, EGF, CSF1, FGF, PDGF, and proteases, MMPs, and secrete components of the extracellular matrix, or ECM. The stromal reaction promotes tumor proliferation and differentiation, suppression of immune rejection, and tumor cell apoptosis, invasion and commitment to metastasis. C. If cancer-associated fibroblasts, CSF1 colony stimulating factor, CXCL5, CXC, motif chemokine, ECM extracellular matrix, or epidermal growth factor, EGF, FGF fibroblast growth factor, MSC mesenchymal stem cells, MMP matrix, metalloproteinases, natural killer cells. Our platelet-derived growth factor, our TAM, tumor-associated macrophage, our TGF-beta, tumor growth factor beta, T regulatory cells, VEGF, vascular endothelial cell growth factor. So, yeah, a lot going on that will help it survive in the body. Several of the hallmarks enablers are primarily genomic alterations that initiate and maintain development of cancer. These will be discussed first and include sustained proliferation, signaling, evading growth suppression, genomic instability, and replicative immortality. Other hallmark enablers are cellular adaptations secondary to genomic changes and include inducing angiogenesis and reprogramming energy metabolism. A third group, tumor resistance to destruction by the host's protective mechanisms, include resisting apoptotic cell death, promoting tumor inflammation and avoiding immune destruction, and last is the culmination of the previous 9, activation, invasion, and metastasis. So our genomic hallmarks include sustained proliferation signaling. The first and foremost hallmark of cancer is uncontrolled cellular proliferation. Normal cells generally only enter proliferative phases in response to growth factors that bind to specific receptors, the receptor tyrosine kinase (RTKs) on the cell surface. The cytoplasmic component of the receptors are associated with signaling molecules that undergo activation and in turn activate intracellular signaling pathways, leading to the induction activation of regulatory factors affecting DNA synthesis, entrance into the cell cycle, and changes in expression of other genes related to cell metabolism for optimal growth. One example is the initiation of cell proliferation by epidermal growth factor, or EGF. Our epidermal growth factor binds and cross-links to epidermal growth factor receptors, or EGFRs, on the cell surface. The cytoplasmic portion of the receptors are tyrosine kinases that attach phosphorus to tyrosine in neighboring proteins, including each other. So, autophosphorylation. Phosphorylation allows the receptor to attach to bridging protein, which links the EGF, or epidermal growth factor, receptors to plasma membrane-associated inactive RAS. RAS is an acronym for rat sarcoma, where it was found originally. Inactivated rat sarcoma is associated with guanine diphosphate, GDP, guanine diphosphate. Association between the EGF receptor and inactive RAS modifies the binding of GDP. So the association between our epidermal growth factor and inactivated rat sarcoma modifies our guanine diphosphate. Okay. Which is then replaced with a guanine triphosphate. Does it give you an extra phosphate from the inactive RAS? GTP activates RAS, which is a GTPase that converts GTP to GDP, during which it can activate signaling pathways, such as the mitogen-activated protein kinase pathways, or MAPK pathway, and the phosphatidylinosytyl 3 kinase pathway, the PI3K pathway. These signaling pathways phosphorylate other cytoplasmic proteins and affect activity and nuclear localization of transcription factors, such as the MYC, the myelocytomatosis viral oncogene homologue, that govern the transcription of cell cycle regulators, such as cyclins, and entrance into cellular proliferation. Proliferation can be discontinued through the pathway by decreased levels of growth factor in the environment or inactivation of signaling pathway components. Ooh, all of that was a mouthful. So if we have growth factor, and we have our growth factor receptor, and we have our bridging protein, but it has part of the inactive rat sarcoma, and that bridging protein activates that RAS, it now becomes an active RAS, and therefore its GTP via the GAP increase and activation by hydrolysis of GTP turns back into a GTP into a GDP. Oh boy, okay. So once the RAS is active, the RAF and MAPK activation of transcription kicks in the MYC and D cyclins, which help its cell cycle progression. It also activates the PI3K via the P10, or with the P10. That turns into AKT, which also pro-growth metabolism, as well as kicks in the mTOR, which increases the protein synthesis, all of which help cellular growth. So pretty much it uses the growth factor to massively produce cell growth, and that's why we have so much proliferation. Makes sense. It uses the resources available to it to regenerate and create more and more and more. The genes that encode components of receptor-mediated pathways designed to regulate normal cellular proliferation are collectively called proto-oncogenes. Cancerous cells characteristically express mutated or overexpressed proto-oncogenes, which are referred to as oncogenes. Oncogenes are independent of normal regulatory mechanism, thus the cell is driven into a state of unregulated constitutive expression of proliferation signals and uncontrolled cell growth. Oncogenes can affect any portion of the growth factor pathways, such as described for EGF. For instance, most growth factors originate from neighboring cells, but some cancers acquire the ability to secrete growth factor that stimulate their own growth, a process known as autocrine stimulation. As described later in this chapter, non-cancerous stromal cells within the tumor are frequently modified to benefit the cancer. In some instances, stromal cells produce excessive growth factor that drive the proliferation of cancer cells. Other cancers increase the expression of RTKs, for example, in breast cancer production of the human epidermal growth factor receptor, or HER2, also known as the epidermal growth factor receptor gene, or ERBB2, is upregulated and is hyperresponsive to low levels of EGF. Some breast and lung cancers are effectively treated by inhibitors of HER2 and other EGF receptors that block this pathway. Oncogenes may lead to constitutive activation of signal cascade from the cell surface receptor to the nucleus. Up to a third of all cancers have an activating mutation in the RAS gene, resulting in a continuous cell growth signal even when growth factors are missing. Other mutations in the EGF receptor pathway include excessive proliferation signaling by hyperactivating or hyperactivation of the P13 kinase. Several types of genetic events can activate oncogenes. A point mutation that is frequently observed in lung cancer results in continuous activation of the EGF receptor tyrosine kinase. A point mutation in the RAS gene converts it from a regulated photo-oncogene to an unregulated oncogene. Activating point mutations in RAS are found in many cancers, especially pancreatic and colorectal cancer. Specialized tests such as a direct DNA sequencing can detect such point mutations in clinical samples. Translocations can activate oncogenes in one or two distinct mechanisms. First, a translocation can cause excess and inappropriate production of proliferation factor. One of the best examples is the T8 or 14 translocation found in many Burkitt lymphomas. Designates a chromosome that has a piece of chromosome 8 fused to a piece of chromosome 14. Burkitt lymphoma is an aggressive cancer of B lymphocytes. The MYC proto-oncogene found on chromosome 8 is normally activated at low levels in proliferating lymphocyte and is inactivated in mature lymphocytes. If the T814 translocation occurs, reciprocal translocation, T, or exchange of genetic material between chromosomes, designated T8 for chromosome 8 and 14 for chromosome 14, the MYZ gene is aberrantly placed under the control of a B-cell immunoglobulin gene, or Ig, present on chromosome 14. The Ig gene is very active in maturing B lymphocytes. The T814 translocation alters the control of MYC. Its normal low-level expression is switched to high-level, as directed by the Ig gene promoter. Hyperproduction of MYC protein drives proliferation and blocks off its differentiation. Whew, okay. So because they, I guess, bounce off each other so much, it causes a trigger in its proliferation. A second chromosome translocation can lead to a production of novel proteins with growth-promoting properties. In chronic myeloid leukemia, or CML, a specific chromosome translocation is almost always present. This translocation was first identified in association with CML in Philadelphia in the 1960s and is often referred to as the Philadelphia chromosome. Translocation fuses two chromosomes in the middle of two different genes, the BCR, breakpoint cluster region, on chromosome 9, and ABL, the Abelson gene, on chromosome 22. The result is production of the BCR-ABL fusion protein containing the first half of BCR and the second half of ABL, a non-receptor tyrosine kinase. BCR-ABL is an is an unregulated protein tyrosine kinase that promotes growth of myeloid cells. Imatinib, a drug that specifically targets the tyrosine kinase, represents the first successful chemotherapy targeted against the product of a specific oncogenic mutation. Imatinib and related tyrosine kinase inhibitors, TKIs, are highly effective in the treatment of CML, and because of their specificity, lack the toxic side effects noted with nonspecific anti-cancer drugs. However, imatinib is not effective in cancers that do not have the T922 translocation or related mutations. In modern personalized cancer therapy, knowledge of the specific genetic alterations can dictate the optimal drugs for the individual. Oncogenes also may be activated by gene amplification. Gene amplification results in increased expression of an oncogene, or in some cases drug resistance genes. The NMYC oncogene, a member of the MYC family, is amplified to 25% of childhood neuroblastomas and confers a poor prognosis. The HER2 gene, or ERBB2, is amplified in 20% of breast cancers. The NMYC gene amplification in neuroblastoma. Picture A, the NMYC is present on the chromosome 2. Then it becomes amplified and is seen either in extrachromosomal couple of minutes or as a chromosome. It's detected in human neuroblastoma cells using a technique called the FISH, the fluorescent in situ hybridization. So B, a single pair of NMYC genes is detected in normal cells in a low-grade neuroblastoma. But in picture C, multiple amplified copies of the NMYC genes are detected, and in some cases of neuroblastomas, amplification of this is strongly associated with poor prognosis. That's unfortunate. Thanks to the university or the School of Medicine in Salt Lake City. So if we see 2, we're in the green. If we see more than 2, we're in the deep dark red. Evading growth suppressors. Uncontrolled cancer cells proliferation also is related to the inactivation of tumor suppression gene. Tumor suppressor genes normally regulate the cell cycle, inhibit proliferation resulting from the growth signal, stop cell division when cells are damaged, and prevent mutations. Hence, they also have been referred to as anti-oncogenes. Whereas oncogenes are activated in cancers, tumor suppressors must be inactivated to allow cancer to occur. Right. You have to turn off the killer portion of it to get the life portion. Whereas if we turn up the killer portion, it takes off the life portion. So we actually want tumor suppressor genes to ensure that our cells go through natural apoptosis. So whereas oncogenes are activated in cancers, tumor suppressors must be inactivated to allow cancer to occur. A single genetic event can activate an oncogene because it can act in a dominant manner in the cell. However, each individual has 2 copies of each tumor suppressor gene, one from each parent. Both copies must be inactivated before mutations are necessary. And here is a series of comparisons of cancer gene types. Caretaker, DNA and chromosome stability. So the mutation would be chromosome instability and increased rate of mutation. dominant oncogenes, its normal function is to encode proteins that promote growth, but the mutation is an overexpression or amplification causing gain of function. Whereas the gene tumor suppression or recessive oncogene, its function encodes proteins that inhibit proliferation and prevent or repair mutation. Its mutative effects require loss of function of both alleles to increase cancer cells. A non-mutant state referred to as a proto-oncogene. Oh boy. A prototypical tumor suppressive gene is the retinoblastoma, or RB gene. Normal cells receive diverse anti-growth signal from their normal environment, contact with other cells, with basement membranes, and with some soluble factors normally signal cells to stop proliferating. Tumor suppressor genes, such as RB, monitor anti-growth cellular signal and block activation of the growth division phase in the cell cycle. Thus, mutations in RB lead to persistent cell growth. Anti-proliferative activity of RB depend on the degree of protein phosphorylation. Low levels of phosphorylation or hypophosphorylation result in the RB binding to and inhibiting transcription factors that regulate gene controlling passage through the cell cycle. Growth factor regulated kinases increase phosphorylation or hyperphosphorylation and inactivation of RB. Okay, so if we have a low growth factor regulation, there is an increase in RB. If there is a high level of phosphorylation, then there is a low RB. A variety of genetic mutations in cancers also inactivate RB, resulting in unregulated and continuous cellular proliferation. RB is mutated in childhood retinoblastoma and in many lung, breast, and bone cancers as well. The RB gene resides on chromosome 13 in a region referred to as Q14, so 13q14. Most individuals with RB mutations have a subtle mutation, such as a point mutation in one allele. The RB gene is in the other chromosome may be inactivated through the loss of the 13q14 region of the epigenetic mechanism. Another classic tumor suppressor gene is p53, tumor suppressor gene, or TP53. The protein p53 has been called the guardian of the genome. The TP53 monitor intracellular signals related to stress and activates caretaker genes, genes that are responsible for the maintenance of genomic integrity. Many types of cellular stress, anoxia, oncogene expression, nuclear damage, produce intracellular signals, levels of nucleotides and glucose, degree of oxygenation, DNA damage, and other indicators of cellular abnormalities. Detectable by the p53, normally p53 is an inactive complex with inhibitor molecules. Stress activates kinase and that phosphorylate p53 into an active suppressor of cell division and activator of caretaker genes. Caretaker genes encode proteins that are involved in repairing damaged DNA, such as with errors in DNA replication, mutations caused by ultraviolet or ionizing radiation, and mutations caused by chemicals and drugs. The p53 protein also controls initiation of cellular senescence or apoptosis and suppresses cell division until DNA repair is complete or other effects of stress are corrected. If not corrected, the cell enters senescence or apoptosis, thus preventing DNA damage and mutation. Loss of function of TP53 or caretaker gene leads to an increased rates of cancer. So I guess that's why it's called the Guardian. It's pretty much our big line of defense to make sure that cancer cannot mutate properly. Because of inactivation of tumor suppressor genes require at least 2 mutations, one in each allele, a single mutation in the germline cells, sperm or egg, result in the transmission of cancer-causing genes from one generation to the next, producing families with a high risk of specific cancers. These inherited mutations that predispose to cancer are almost invariably in tumor suppressor genes because only a single additional mutation is needed to inactivate completely the tumor suppressor gene. An example of increased risk for cancer that can be inherited is the familial form of retinoblastoma or retinoblastoma. A mutation in one RB allele is inherited so that only one additional mutation in the normal allele will lead to cancer. Approximately half of children with retinoblastoma have an inheritable form, and most will develop tumors in both eyes. Bilateral retinoblastoma, also Li-Fraumeni syndrome, is a very rare inheritable loss of function mutation in TP53 in one allele, resulting in a 25-fold increase of developing malignancy at early age, younger than 50. These malignancies may include breast cancer, brain tumors, acute leukemia, soft tissue sarcomas, bone sarcomas, and adrenal cortical carcinomas. Other familial cancers with inheritable mutations in tumor suppressor genes include Wilms tumor, a childhood cancer of the kidneys, neurofibromatosis and familial polyposis, coli or adenomas of the colon, characterization of cancer-causing genes and other genetic factors help identify individuals prone to developing cancers and contribute to the understanding of the sporadic cancers. Individuals known to carry mutations in tumor suppressor genes are offered targeted cancer screening to facilitate early cancer detection and therapy. All right. The genomic instability refers to an increased tendency of alteration, mutability, in genome during the life cycle of cells. Inherited and acquired mutations in caretaker genes that protect the integrity of the genome and DNA repair increase the level of genomic instability and risk for developing cancers. Acquired mutations in guardians of the genome, such as TP53, that detect DNA damage and activate repair mechanism resulting in the increased accumulation of mutation. Xeroderma pigmentosum is a defect in the repair of DNA pyrimidine dimers created by UV light that increase the risk for skin cancer. Hereditary non-polyposis colorectal cancer resulting from an inherited defect in the repairing DNA base pair, mismatches that occur occasionally during DNA replication. Affected individuals have an increased rate of small insertions and deletions in DNA, leading to a high rate of colon and other cancer. Some inherited mutations threaten the integrity of the entire chromosome. Bloom syndrome, caused by the mutation in a DNA helicase, presents with an increased risk of several forms of cancer, and those with Fanconi aplastic anemia, caused by loss of function by repairing DNA double-strand breaks, have a particular increased risk of acute myelogenous leukemias. These examples are autosomal recessive disorders in which affected individuals demonstrate marked chromosomal instability. Genomic instability also may result from increased epigenetic silencing or modulation of gene function. Many cancers have increased methylation of DNA in the promoter region of tumor suppressor genes. They also have associated changes in the modification of histones in the chromatin, often correlated with methylation of DNA. These changes alter the promoter regions of genes, leading to their silencing or altered gene expression. Changes in gene regulation can also affect not just single genes, but also entire intracellular signaling networks. Gene expression networks can be regulated by changes in microRNA or miRNAs and other non-coding RNAs and cRNAs. MIRs regulate diverse signaling pathways. The MIRs that stimulate cancer development and progression are termed oncomiRs. Mirs decrease the stability and expression of genes by pairing with mRNA. So, mutations in the BRCA1 and BRCA2, the breast cancer 1 and 2 early onset genes, respectively, are currently of clinical importance. Both are tumor suppressors and caretaker genes that repair double-stranded DNA breaks. Inherited mutations in either gene greatly increase the risk for a variety of tumors, especially breast cancer, in both women and men, and ovarian or prostate cancers. Approximately 12% of the women generally will develop breast cancer within their lifetime, whereas about 60% of women with a high risk of BRCA1 mutation and 45% with a BRCA2 mutation will develop cancer by age 70. Ovarian cancer occurs in approximately 1.4% of the general population, but about 39% of women with an inherited mutation of BRCA1 and about 15% with the mutation of BRCA2 will develop ovarian cancer by age 70. At-risk women are currently offered prophylactic surgery to reduce the risk of cancer. In addition to specific gene mutations and abnormal epigenetic silencing, chromosome instability also appears to be increased in malignant cells, resulting in a high rate of chromosome loss, as well as loss of heterozygosity and chromosome amplification. The underlying mechanism of this instability is not clear, but may be caused by malfunctions in the cellular machinery that regulates chromosome segregation at mitosis. So, enabling the replication immortality. A hallmark of cancer cells is their immortality, in that they seem to have an unlimited lifespan and will continue to divide for years under appropriate laboratory conditions. One of the most common used laboratory cell lines, such as the HeLa cells, were derived from a cervical cancer specimen obtained in 1951 that continues to grow and divide in laboratories around the world. Most normal cells are not immortal and can divide only a limited number of times, known as the Hayflick limit, before they either enter senescence, cease dividing, or enter crisis, apoptosis, and die. One major block to unlimited cell division, or immortality, is the size of the specialized structure, called the telomere. Telomeres are protective ends, or caps, of repeating hexanonucleotides on each chromosome and are placed and maintained by a specialized enzyme called telomerase. As one might expect, telomerase is usually active only in germ cells, such as the ovaries and testes, and in stem cells. All other cells of the body lack telomerase activity. Therefore, when non-germ cells begin to proliferate abnormally, the telomere caps shorten with each cell division. Short telomeres normal signal to cease cell division. Oh my god. Short telomeres normally signal the cell to cease cell division. Got it. So the less there is, the shorter its lifespan, or its ability to replicate and duplicate. If the telomeres become critically small, the chromosomes become unstable and fragment and the cell dies. So, I guess that's one way to end cancer, but normally we don't outlive that. Cancer cells are very heterogeneous, and many cells die as the cancer develops. When they reach a critical age, most cancer cells activate telomerase to restore and maintain their telomeres, thereby allowing continuous division. The trigger for re-expression of telomerase activity remains unclear, but seems to require expression of specific oncogenes such as RAS and MYC. The loss of function of certain tumor suppressor molecules such as the p53 and Rb, telomerase activity is restored in about 90% of cancers. The remaining cancers appear to recruit or originate from stem cells. Becoming cancer stem appears to recruit. Wait, what? Sorry, lost my spot. Becoming cancer stem cells that maintain levels of telomerase activity characteristically found in somatic stem cells. Because telomerase is specifically activated in cancer cells and potentially in cancer stem cells, it is an attractive therapeutic target. So cellular adaptations, inducing angiogenesis. A major component of wound healing is the process of establishing new blood vessels within the tissue undergoing repair, called neovascularization or angiogenesis. Access to a blood supply also is obligatory to the growth and spread of cancer. Without a blood supply to deliver oxygen and nutrients, growth of a tumor is limited to about a millimeter in diameter. Angiogenic factors and angiogenic inhibitors normally control development of new vessels. In cancerous tumors, several mechanisms increase and maintain secretion of angiogenic factors by the cancer cells, as well as prevent release of angiogenic inhibitors. Hypoxia-inducible factor 1 alpha, or HIF-1 alpha, an oxygen-sensitive transcription factor, is a major regulator of angiogenesis in normal tissue. Hypoxia-induced factor 1 alpha is stabilized under hypoxic conditions and induces expression of pro-angiogenic factors, such as the vascular endothelial growth factor, or VEGF, and basic fibroblast factor, BFGF. Inactivation of tumor suppressor genes, RP53 Guardian, or increased expression of oncogenes, the HER2, leads to increased expression of the hypoxia-induced factor 1 alpha regulated angiogenic factors and increased vascularization. This increased expression of HIV-1A or alpha also is related to increased resistance to chemotherapy, increased tumor cell glycolysis, increased metastasis, and a poor prognosis. These effects may likely occur through the autocrine mechanism by which VEGF activates tumor-associated VEGF receptors. For instance, in some soft tissue sarcomas, VEGF induces increased expression of anti-apoptotic proteins and activation of intracellular survival signal pathways. The use of angiogenic inhibitors targeting VEGF signaling can inhibit angiogenesis and diminish tumor growth. Other routes of angiogenic factor induction includes mutation in cancer oncogenes that increase transcription of VEGF by cancer cells. Most cells in the tumor microenvironment also secrete VEGF, including tumor-infiltrating monocytes, endothelial cells, adipocytes, and cancer-associated fibroblasts. Angiogenesis inhibitors, such as thrombospondin 1, TSP1, normally bind to cellular surface receptors or inflammatory cells and negatively regulate angiogenesis in wound healing and tissue remodeling. The expression of angiogenesis inhibitors is under the control of our guardian P53, which is suppressed in cancer cells, thus diminishing the control of stromal inflammatory cell secretion of angiogenic factors. Cancer cells and stromal cells may increase production of matrix metalloproteinases, or MMP9. MMPs are zinc-dependent proteases that digest the surrounding extracellular matrix, or ECM. The extracellular matrix, ECM, contains stored latent or inactive forms of some angiogenic factors. The BFGF transforming growth factor beta, or TGF-beta, the matrix metalloproteinases activate the stored forms of functional angiogenic factors. Wow. So, if our circulating precursor endothelial cells jump in and our BFGFR, our blood vessel, in the blood vessel, there is a whole lot of jumping back and forth, mostly from the tumor, the VEGF and the VEGFR, our PDGF or PDGFRs, those receptor sites, and then finally our BFGF connect to their respective receptor sites. So the tumor-induced angiogenesis, malignant tumor, oh no, make this a little large enough so that we all can see it. Alright, I'll scroll down just a little bit so we can read it all. The tumor-induced angiogenesis. Our malignant tumor secretes angiogenic factors and tissue remodeling matrix metalloproteinases, those MMPs that we were just talking about, that actively induce formation of new blood vessels. These new blood vessels are formed from both local endothelial cells and circulating precursor cells, recruited from the bone marrow. Circulating platelets can also release regulatory proteins into the tumor that BFGF and its associated receptor site, the BFGFR, basic fibroblast growth factor and its receptor, and respectively the PDGF and PDGF receptor site. Platelet-derived growth factor and its receptor, respectively, the VEGF and VEGFR, vascular endothelial growth factor and its receptor. So, from tumor to vasculature. Quite interesting. The vessel formed within tumors differ from those in healthy tissue. They originate from endothelial sprouting, from existing capillaries, and irregular branching. rather than regular branching seen in healthy tissue, the interendothelial cell contrast is less tight, so the vessels are more porous and prone to hemorrhage, as well as allowing passage of tumor into cells and the vascular system. And our last little section, I believe, is our reprogramming energy metabolism. Cancer cells live in distinct environment from normal cells and have different nutritional requirements from non-proliferating cells. The successful cancer cell divides rapidly, with the consequent requirements for the building blocks to conduct new cells. Non-malignant cells, in the presence of adequate oxygen, normally generate adenosine triphosphate, or ATP, by mitochondrial oxidative phosphorylation, or OXPHOS, generating 36 ATP molecules from each glucose molecule that is broken down to water and carbon dioxide. In the absence of sufficient oxygen, or hypoxia, normal cells perform glycolysis, or anaerobic glycosis, generating only 2 ATP molecules per molecule of glucose, with lactic acid and pyruvate as its byproduct, which we've known since our biology. Even in the presence of adequate oxygen, cancer cells may not use the mitochondrial oxidative phosphorylation, or oxygen phosphorylation, but are reprogrammed by glycolysis, or the Warburg effect. The Warburg effect is the use of glycolysis under normal oxygen conditions, hence the name aerobic glycolysis. Although aerobic glycolysis was postulated to arise from cancer-specific mitochondrial dysfunction, it is now apparent that this is instead a highly regulated and beneficial adaptation for cancer cells. The shift from oxygen phosphorylation to glycolysis also allows lactate and other products of the glycolysis to be used for more efficient production of lipids, nucleosides, amino acids, and other molecular building blocks needed for rapid cell growth. So even if we're under hypoxic events, cancer will thrive. So we have our differentiated tissue. We use our glucose and get our pyruvate or lactate. Which I guess through the mitochondria will create our lactate, oxygen, and CO2. Or we have our hypoxic event, in which our glucose turns into pyruvate and then into lactate. So the proliferation tissue, or tumor, will use that increase or decrease in oxygenated glucose and use pyruvate to give us less oxygen and CO2 and more lactate. But I guess during the anaerobic glycolysis or Warburg effect, we get a negative 4 mole ATP per molecule glucose. Okay, so I guess there is a new model, or reverse Warburg effect. May play a role in certain cancers. Cancers may continue using the oxygen phosphate to generate large amounts of ATP. However, they may also manipulate the cancer-associated fibroblasts, perhaps inducing oxidative stress to undergo aerobic glycolysis and secrete metabolites, such as the lactate and pyruvate, that the cancer cells can use in the citric acid cycle, or Krebs cycle, to feed the oxygen phosphorylation and produce more ATP. A secondary consequence would be the induction of autophagy. Autophagy. That's mispronounced. In the CAFs, cancer-associated fibroblasts, resulting in the consumption of cancer-associated fibroblasts and release of materials needed by the cancer cells in the synthesis of the organelles. Promoters of aerobic glycolysis are activated by oncogenes and mutated tumor suppressor molecules. Upregulation of the GLUT1 or glucose transporter 1 under the control of oncogenes are RAS and MYC and mutant tumor suppressors such as our divine protector TP53 increases transport of glucose into the cytoplasm. These and other oncogenes or mutant tumor suppressor genes inhibit OXPHOS and promote the aerobic glycolytic pathway and related metabolic pathways that support the rapid cancers or the rapid growth of cancers. Clinically, the high glucose utilization of cancer can be exploited for its detection. Fluorodeoxyglucose, FDG, is incorporated into the cells in the same way as glucose, with two key differences. Because it is missing the key hydroxyl group, it cannot be broken down by glycolysis, and thus FDG accumulates in cells. Because it is tagged with an 18F, it can be imaged by a positron emission tomography or PET scan. Small metastatic tumor masses that are consuming huge amounts of glucose can readily be detected with this imaging method, which is why we do PET scans in order to see if you have cancer. So really, when we do a PET scan, what we're really looking for is that positron emission topography. So we're looking for the 18F? Or is it because we see the large amounts of glucose that's being used? Or is it the hydroxyl group that's missing, so it can't be broken down? And we see it via the fluorodeoxyglucose. Interesting. All right, well, we're jumping ahead. We are officially finished with chapter 12. I'm going to jump straight to chapter 13, where we only have a few pages to read there. And by few, it's a couple slides. So, cancer epidemiology. Again, we're not doing the full chapter here. We're only doing the portions required for our reading. So, I'm sure we have some just a couple things to match up here in terms of our questionnaire. So, we'll see. We'll see if we have to stop for anything. But jumping right in. Although cancer arises from a complicated and interacting web of multiple etiologies, avoiding high-risk behaviors and exposure to individual carcinogens, or cancer-causing substances, will prevent many types of cancer. In fact, estimates as high as 80% to 90% for smoking-related cancers, such as lung and oropharyngeal cancer, and as high as 60% of other common lifestyle cancers, such as colorectal and bladder cancer, are preventable. Lifestyle behaviors, dietary and environmental factors, such as exposure to ultraviolet radiation and infections, and occupational exposure contribute to cancer cases and death. In this context, any of the following factors can contribute to the development of cancer. So, some of the key associations with cancer: tobacco, alcohol, indoor and outdoor air pollution, lack of fruits and veggies, processed red meat, lack of fiber, hyperglycemia or hyperinsulinemia, pretty much being a diabetic, being overweight, lack of exercise, post-menopause in women, a series of infections, ionizing radiation, occupational hazards, reproduction, sun exposures and sunbeds, and our biggest one, tobacco. So, lifestyle choices such as smoking, alcohol, nutritional intake, lack of physical exercise, overweight and obesity, infections and sexual practices, environmental conditions, air pollution, sunlight, natural and medical radiation, workplace exposures, and involuntary or unknown exposures, prescribed and illicit medications, socioeconomic factors that affect exposure and susceptibility, and carcinogenic substances present in air, water, and soil. The estimate of environmental factors and their attributable risk for cancers vary. According to the International Agency for Research on Cancer, is part of the World Health Organization. A major goal for the international agency is to identify causes of cancer, and it has evaluated more than 900 candidate carcinogens and placed them in the following groups. Group 1 causes cancer to humans, and Group 2A probably causes cancer to humans, and Group 3 unclassifiable as to the carcinogenesis, its ability to cause cancer in humans, and then finally Group 4 probably not carcinogenic to people. But the fact that they say probably doesn't mean it's not. Because it is difficult to test candidate carcinogens, most are listed as probable, possible, or unknown risk. About 100 chemicals, occupations, physical agents, biologic agents, and other agents are classified as carcinogenic to humans. There's a simple list here, and we can brief through some of it. Apparently, solar radiation can cause some kind of lip cancer, whereas alcohol, tobacco, smoking, any kind of smoke form, whether it's smokeless or smoke-informed, can cause oral cancers. Radiation can cause salivary gland cancers. HPV can cause tonsil cancers. The pharynx again has to do with smoking, HPV, and alcohol, but also from asbestos. I'm not sure what mate drinking is, but mate drinking. Hot printing press. Oh, I think this is like job related. No. Agents with limited evidence in humans. Okay. Tobacco smoke, secondhand smoke. Our nasopharynx via the Epstein-Barr virus, formaldehyde, for some reason Chinese-style salted fish, and wood dust. And then GI upper tract, acetaldehyde associated with consumption of alcohol beverages, digestive, our esophagus, again that same acetaldehyde and alcohol, tobacco, and radiation. Whereas that can be, I guess, done via work or environmentally, dry cleaning, mate drinking. Some pickled vegetables in traditional Asian dishes, the rubber production, and tetrachloroethylene. Big stomach cancer issues is the Epstein-Barr, asbestos, lead compounds, nitrate that's ingested, pickled vegetables, and salted fish. but it's also our H. pylori, rubber production, smoking, radiation. I feel like our common factor here is going to be radiation in pretty much every kind of cancer. Your colon and rectum is again somehow including smoking and tobacco, as well as asbestos, and some kind of schistosoma japonicum. I'm assuming that is our Japanese associated. No, it's not. That was an assumption. The assumption was there. Anal, HIV, and HPV. Liver and bile, aflatoxins, alcohol, estrogen, progesterone, contraceptives. So that's always good to know. Our hepatitis B virus, hep C virus, um, Opisthorchis viverrini, plutonium, because people get in contact with that all the time. Tobacco, smoking, vinyl chloride. So we also have our androgenic steroids, arsenic, the betel quid without tobacco, again HIV, polychlorinated biphenyls, that same schistosoma in Japonicum, trichloroethylene, and radiation. Our gallbladder cancers are usually due to thorium-232 and its decay products. Pancreatic cancer is said to be due to smoking and smokeless cancers, but also alcoholic beverages, again at thorium-232 and radiation. And GI unspecified, but they're calling it radioiodines. Respiratory are isopropyl alcohol production, leather dust, nickel, radium, smoking, wood, so carpentry was a big issue, and formaldehyde, and textile manufacturing. Our larynx, acid mists, strong inorganic alcoholic beverages, asbestos, smoking, but also HPV, mate drinking, rubber production, sulfur mustard, tobacco, whether it's primary or secondhand. Lung cancers, aluminum production, arsenic, coal, cadmium, coke, hematidine mining, iron, steel, nickel, painting, plutonium, radon, rubbers, silica dust, soot, sulfur, tobacco, radiation. So really, just about anything can cause lung cancer. Acid mists, art glass production, biomass fuel such as wood, vitamins, carbon electrode, cobalt, engine exhaust, frying, insecticides, printing processes, tetrachlorodibenzoes, welding fumes, bone cancer due to plutonium, radium, and radiation, and radioiodines. So, all unhealthy. Skin cancers, well, ultraviolet emitting tanning beds, and solar radiation. So, sitting in the sun too long. Who knew that? Other skin malignant neoplasms could also be contributed, though, to arsenic, azathioprine, coal, methoxylene, cyclosporine, mineral oils, shale oils, again that solar radiation and radiation, but also HIV, HPV, nitrogen mustard, petroleum refining exposure, UV emitting tanning devices, and the Merkel cell polymerase, or MCV. There's no way we're reading all that. We're almost at that. Alright. let's look at the big ones. Um, your endothelium or Kaposi sarcomas are normally seen in HIV. Your breast cancer is caused due to alcohol and estrogen-progesterone contraceptives, as well as estrogen-progesterone menopausal therapy. So your contraceptives and your menopause medicine can cause breast cancer, as well as smoking and any kind of shift work that involves your circadian disruption. So women probably shouldn't work at night. Your vulva and vagina can be caused by HIV and HPV. Uterine, again, your contraceptives, HIV, HPV, smoking. Your ovaries can be caused, or ovarian cancer is your estrogen menopausal therapy, asbestos, smoking, and radiation. And any kind of talc-based body powder in perianal use or perineal use. So I'm not sure what kind of talc-based body powder is being used there, but don't do it. Your penis cancers include HPV and HIV, but prostate cancer and testicle cancer can be caused by androgenic steroid abuse, arsenic, cadmium, rubber production industry, your thorium, and X-radiation or X-rays, as well as any kind of diethylstilbestrol exposure. Kidney cancer from smoking or arsenic, but also renal, pelvis, and ureter cancers can be done by smoking. Aristocolic acids, so any kind of plants containing phenacetine, and aristocolic acids, again, same thing. Okay. Urinary bladder from aluminum, arsenic, aramine, benzidine, magenta, cyclophosphamide, so pretty much our coal and tar pitch. Ooh, interesting. Urinary bladder cancer can come from coffee. Better put my coffee down then. Engine exhaust, especially from diesel. Your hairdressers and barbers have an occupational exposure. Interesting. I'm curious how that correlates. Why they would get a higher sense of urinary bladder cancer. But also the printing process, soot, and textile manufacturing. Ooh, actually the list goes on. Painting, rubber production, tobacco smoking can also cause urinary bladder cancers. Eye cancer from HIV and solar radiation, UV and welding. So that very high light radiation. Brain and central nervous cancers from X-rays. Your thyroid cancers from X-rays. Your leukemias is a long, long list. So let's pick out the big ones. Benzene, cyclosporins, Epstein-Barr, formaldehyde, H. pylori, Hep C, HIV, HPV, Kaposi sarcomas, rubber production, your phosphorus, smoking, X-rays, lots of reasons. And then if you've got cancers in multiple sites, well, it probably is a fission production. Cyclosporine, X-rays, and all cancers combined have a type 2, 3, 7, and 8 tetrachlorodibenzoparadioxin. So, whew, you name it, it can cause cancer somewhere. As I drink my cup of coffee. Alright, so genetics, epigenetic, and tissue. Cancers are caused by environmental, lifestyle, and genetic factors. Cancer is driven by genetic alteration and changes in epigenetic regulation. Critical cancers include oncogenes and tumor suppressor genes. These genes become altered by a combination of genetic and epigenetic alterations to drive cancer progression. Critical genes for cancer regulate when cells grow, divide, differentiate, or die. A subclass of tumor suppressor genes help maintain genome integrity. Although driver and passenger genetic alterations are key, a wealth of data now indicate the importance of epigenetic processes, especially those with resultant gene silencing. So, not broken, but mute, of key regulatory genes. Different combinations of mutations and epigenetic changes are found in different types of cancers. Neoplastic cells undergo a dynamic and reversible transition between multiple phenotypic states, including the epithelial mesenchymal plasticity in cancer. This plasticity is enabled to change in epigenetic regulation. Interacting factors that influence cancer risk include detoxifying enzymes, DNA repair genes, immune and inflammation systems, and the cell's immediate environment. The biologic environment surrounding cells include metabolic and hormonal factors. For example, excess estrogen production, inflammation, and disordered glucose and lipid metabolism. The cell's biologic environment is modified by metabolic requirement, physical activity, infections, nutrition, occupational carcinogens, air pollution, and many other environmental factors. Investigators are challenged to connect the complex web between genotype, phenotype, and environment lifestyle factors to understand a person's chance of developing cancer. Cancer development and progression involve the tissue microenvironment or stroma. The microenvironment participates in a complex signaling process that facilitates tumor promotion and metastasis because stromal tissue has various immune cells that can promote inflammation. Extensive clinical and experimental evidence shows that infiltrating immune cells cause chronic inflammation and therefore create a permissive tumor progressive environment. Chronic inflammation can also precede and presumably initiate malignant change, as for example in the inflammation-induced colon cancer. It is well documented that chronic inflammation induced by bacteria, viruses, autoimmune processes, and toxins promote common types of cancer, including colon, liver, and lung cancer. Inflammation can be caused by numerous environmental factors. For example, inhaling tobacco, asbestos, or fine particles in the air from diesel engine exhaust and industrial sources. These sources are major factors in lung and respiratory tract cancers. Cancer development in the presence of chronic inflammation involves the continuous presence of cytokines, chemokines, reactive oxygen species, or ROS, oncogenes, cyclooxygenase 2, or COX2, 5-lipo or lipooxygenase, or 5-LOX, and matrix metalloproteinases, MMPs, as well as the activation of essential transcription factors, such as nuclear factor Kb or NF-kB. So the incidence and mortality trend of cancer. Cancer is reported to become a major cause of morbidity and mortality in the coming decades in all regions of the world. According to a report on Globocan, an estimated 14.1 million new cancer cases and 8.2 million cancer deaths were reported, and 32.6 million people were found to be living with cancer. Diagnosed in the past 5 years worldwide, back in 2012. The age standardized rate for all cancers, excluding non-melanoma skin cancer for men and women combined, was 182 per 100,000. The rate was higher for men, 205 per 100,000, than women, 165. The highest cancer rate for men and women together was found in Denmark, with 338 people per 100,000 being diagnosed in 2012. The standard rate was at least 300 per 100,000 for 9 countries: Denmark, France, Australia, Belgium, Norway, the United States, Ireland, the Republic of Korea, and the Netherlands. The top countries in the top 10 come from Europe, Oceania, North America, and Asia. In 2016, annual report to the nation, the National Cancer Institute, the American Cancer Society, the CDC, and the North American Association of Central Cancer Registries collaborated to provide updates on cancer incidence, death rates, and trends in these rates for United States from 2003 to 2012. The overall incidence rates, new cases per 100,000 persons, decreased for men and stayed about the same for women. During this period, 7 of the 17 more common cancers in men showed a decrease in incidence, including colorectal, lung and bronchial, prostate, stomach, larynx, bladder, and brain cancer. 6 of the more common cancers in women showed a decrease in incidence, including cancers of the colorectum, the cervix, lung, the bronchus, bladder, ovary, and stomach. Importantly, the decline in the incidence of lung cancer corresponded with a decrease in tobacco use. During this same period, cancer incidence increased among people ages 0 to 19. The annual reports included a special section on liver cancer. In all racial and ethnic populations, men had about a 3-fold higher liver cancer incidence than that of women. In 2012, the most recent year for which data is available, 28,012 people were diagnosed with liver cancer in the United States, but that was excluding Nevada. Of this total, 20,207 were men and only 7,805 were women. Liver cancer incidence rates were higher among people born between 1945 and 1965 than among those born in other periods because of the higher rates of hepatitis C infection. Among persons born in the cohort, HCV infection was highest among non-Hispanic whites, non-Hispanic blacks, and Hispanics. Cirrhosis or scarring of the liver also is a precursor to liver cancer. Risk factors for cirrhosis include a history of liver disease, history of heavy alcohol use, and some rare genetic disorders. In the United States, alcohol overuse contributes to about 8 to 16% of liver cancer deaths. Researchers found continued decline in cancer death rates or mortality rates for men, women, and children. Death caused by liver cancer, however, increased at its highest rate of all reported cancer sites, and liver cancer incidence rates increased sharply. The mortality rates for men from liver cancer were more than double the rate of those for women, with 15,563 deaths among the men and 7,409 deaths among women. So, what does that mean in utero and early life conditions? From studies of the etiology of certain cancers, it is widely accepted that a long latency period precedes the onset of adult cancers. Accumulating data suggests early life events influence later susceptibility to chronic diseases. Development plasticity is the degree to which an organism develops as contingent on its environment. Specifically, the development origins hypothesis postulates that nutrition and other environmental factors affect cellular pathways during gestation, enabling a single genotype to produce a broad range of adult phenotypes. Plasticity refers to the ability of genes to organize physiologically or structurally in response to environmental conditions during fetal development. The hypothesis also postulates that persistent epigenetic adaptations that occur early in development in response to maternal and paternal nutrition and the environment are associated with increased susceptibility to cancer and other adult onset chronic diseases. Throughout in utero development, the placenta plays a major role in controlling growth and development. Because the placenta is a regulator of the intrauterine environment and can be influenced by exposure through pregnancy, research is being done by DNA methylation linking environment cues to placental pathologies and adult life. The Dutch famine birth cohort is a well-known study of the effects of prenatal undernutrition in humans. Undernutrition was linked to increased heart disease, metabolic disorders, and a possible link with breast cancer decades later. Early versus late undernutrition in pregnancy indicates that the first trimester of pregnancy is particularly vulnerable to disease. Outcomes in adulthood. A striking experiment in mice demonstrated how extra vitamin doses during pregnancy and the mother's diet changed the fur color of her pups. This was the first study to show maternal nutrition and subsequent phenotype changes. The nutrients B12, folic acid, choline, and betaine silenced the gene that rendered mice fat and yellow, but did not alter the DNA sequence. Silencing or switching the gene off linked prenatal diet to such diseases as diabetes, obesity, and cancer. More research is needed to understand nutrition in pregnancy and child vulnerabilities later in life. But there is an ecosystem society population behavior that have an internal environment or external environment, genes, cells, organs, and physiology. The epigenetics of a baby. Perhaps one of the best examples of early life events and future cancer is the chemical exposure to diethylstilbestrose, or DES, a synthetic estrogen. This medication was prescribed between 1938 and 1971 to attempt to prevent multiple pregnancy-related problems, such as miscarriage, premature birth, and abnormal bleeding. By the 1950s, it became clear that DES interfered with the development of the reproductive system in the fetus and did not prevent miscarriage. Recent data suggests that the DES-associated increase in clear cell adenocarcinoma is elevated throughout the women's reproductive years. Other studies have revealed that daughters of women who took DES during pregnancy may have a slight increased risk of breast cancer before age 40. For every 1,000 DES-exposed women ages 45 to 49, it is estimated that 4 will be diagnosed with breast cancer. Research from animal studies have demonstrated a relationship between DES exposure and an increased rate of a rare type of testicular cancer, or rete testes, and prostate cancer. In terms of utero exposures, testicular cancer has been linked to exposure to abnormal levels of estrogen, and testicular cancer is a risk factor for men with undescended testicles, a factor in some studies correlated with DES exposure. However, studies in humans determining the risk of testicular or prostate cancer and DES exposure are unclear and is continuing. Investigators from a recent study reported early life exposure to endocrine disrupting chemicals, or EDCs, including DES, can increase the incidence, multiplicity, and overall size of uterine fibroids in the Eker rat model. In summary, epidemiology and epidemiologic and animal studies reveal that small changes in the development environment can alter phenotypic changes, resulting in individual response in adulthood. Continuing evidence indicates that epigenetic mechanisms are responsible for tissue-specific gene expression during cellular differentiation, and that these mechanisms modulate development phenotypic changes. The phenotypic effects of epigenetic modifications during development may need long latency periods, such as in cancer, thus manifesting later in life. In addition, epigenetic effects may help explain transgenerational effects. So we can have a multi-generational phenotype that has a direct exposure, which is simultaneously exposed of multiple generations to an environmental factor, whereas we can have a transgenerational phenotype with an initial germline exposure, and the transgenerational phenotype is transmitted to future generations through that germline inheritance. Bless you. We also can have a somatic versus germ-like inheritance. The somatic cell's biological response is critical for adult-onset diseases in exposed individuals, not transmitted to future generations, as transgenerational effect, whereas germ cells have a biological response that allow the transmission between generations, promoting transgenerational phenotypes. Lifestyle habits are modifiable, and changing them can translate to reduced cancer risk and prevention of cancer. Examples of environmental factors on cancer risk are abundant. A critical question is whether individuals are intervening early enough in life to achieve cancer prevention. Protective measures in very young people, for example, avoiding sun exposure during peak hours, 10 a.m. to 3 p.m., covering the skin whenever possible, increasing physical exercise, and avoiding high-risk sexual practices will reduce cancer incidence. Understanding that it takes years to develop tumors, cancer prevention beginning early in life will help reduce the burden of cancer on individuals and society. So I think this is where we close our portion of cancer epidemiology. We will take a quick peek here at the normal stem cell. New research has demonstrated how cancers can start in stem cells. So when needed, for example, to repair damage, they replicate to make more stem cells or other cells the body needs. The DNA mistakes in stem cells, sometimes stem cells acquire random mistakes, I'm assuming that's these little dots here, in their DNA as they replicate. Things in the environment can increase DNA mistakes, such as smoking, alcohol, your diet, sun, obesity. So now we have more issues in our stem cells. So what happens to stem cells with DNA mistakes? Well, if the stem cell with DNA mistakes are sleeping, no cancer develops. But if the stem cell with DNA mistakes are replicating, cancer develops. So we have an extrinsic factor, such as tissue damage, and selective mutagenesis. We can have tissue repair, but the evasion of the immune surveillance will cause a propagation of mutated stem cells and progeny, which is our cancer. All right. So I thank you all for watching, for being part of the learning stream here, and we will continue with stress and disease on chapter 11. So we're going a little bit backwards, but I wanted to hit cancer first today. So we'll get back to that momentarily. So we'll see you